talk about how we actually capture the E. coli. The first step of this process is the synthesis of this synthetic amino acid. We can make it in 5 steps. And basically, what we want to do is have this boronic acid handle at the top. Here's the amino acid component. And through peptidoglycan remodeling in E. coli, which occurs faster than other species that might be present, we basically get E. coli that are now labeled with this boronic acid. We can then synthesize this carbazide-containing thiol, shown here is this carbazide group. We can immobilize that on a gold electrode such that it reacts with the boronic acid if it's present. So if we have E. coli that incorporate this boronic acid, they'll react with our surface, and we get E. coli that are covalently attached to our electrode. We can see this by scanning electron microscopy. So shown here are images of our electrode surfaces. We have a bare electrode with boronic acid modified E. coli, and you can see we see no background binding. Similarly, if we have a carbazide modified electrode and E. coli that were not fed that boronic acid, we don't see any background binding. And it's only when we have a carbazide electrode with boronic acid E. coli that we see those E. coli tethered. You can see them attached throughout the electrode surface. But obviously, scanning electron microscopy is not ideal for point of care detection. So we wanted to be able to detect those cells that are attached to our electrode electrochemically. And one of the ways we can do that in a label-free manner is using impedance spectroscopy. So impedance is fundamentally resistance to AC current. And when we think about impedance, we can think about incorporating a redox-active molecule in solutions such as ferrocyanide, ferrocyanide, and looking at the impedance response to that molecule. And there are 2 components we can measure from this. We can look at the electrolyte or diffusive resistance, which is basically just resistance from these molecules to diffusion in solution. And we have the interfacial or charge transfer resistance, which is what we care about. And fundamentally, that's basically just telling you how much of your surface is blocked. And what the impedance data looks like is when we plot it, we have the real impedance on the x-axis here, imaginary on the y, and we can fit this data to an equivalent circuit and extrapolate usable parameters. So from this, we can pull out that charge transfer resistance as a single value. And what that means in terms of our data is it's basically just the diameter of this arc. So qualitatively, you can analyze a lot of this data by just looking at the diameter changes. And so what we would expect to see here is that for an electrode that has no cells bound, we see this small arc corresponding to a small charge transfer resistance. As we add more cells, we're going to see this increase in that interfacial resistance and see that increase plotted here. So does that make sense to everybody about what we're looking at for impedance data? Excellent. So this is what real data looks like. For our system, where we have the E. coli modified with boronic acid, and we bind it to carbazide modified electrodes, we can see that the dotted arcs here are our raw data for titrating in cells ranging from 10 to the second CFU, or colony forming units, it's basically cells per mil, all the way to 10 to the 7th. And I also want to point out our controls here. So if we have no cells, this is basically just our background. And as we saw by SEM, if we have no boronic acid added to those cells, we see no background binding, and this is confirmed by our electrochemical measurement. Importantly, if we add Staph aureus that have been exposed to that boronic acid, even at very high concentrations of cells, 10 to the 7th CFU per mil, we see no response shown here. And this is because of the rate of peptidoglycan remodeling. That's how we get specificity with this sensor. So as you can see, we get this very large detection range, but this data is hard to interpret in the sport, right? So what we can do is fit that data to those model circuits I showed before, and that is what those solid lines are. And from that, we can basically pull out the charge transfer resistance and plot that as a function of the number of cells. And as we can see, for E. coli detection in both buffer and synthetic urine, we have an extremely large range of detection between 10 to the 2nd and 10 to the 7th cells per mil. And our limit of detection for this is about 15 cells per mil, which is 2 orders of magnitude below the infectious dose of these cells. Additionally, if we add E. coli to milk, which is often very challenging to detect from because the number of proteins present, we still maintain our ability to detect this over several orders of magnitude. And as we would expect for Staph aureus And for unlabeled E. coli, we see no background signal. So we can do this in a lab environment, but can we do this from real samples? So next, what we sought to do is to compare our technique to the current gold standard for E. coli detection. For those of you that are unfamiliar with this technique, it involves plating cells on agar plates and then waiting for them to grow, often for several days. And this is really state of the art. So we wanted to see how well our electrochemical method aligned with that. So shown in purple here is our electrochemical measurement, and shown in gray is colony counting, which is just literally physically counting the number of cell colonies on a plate. So as you can see, we were able to get eggs, romaine lettuce, and spinach, as well as chicken from a local farmer's market in Northern California. And we were able to get a feline urine sample that was confirmed to have an E. coli and UTI from the vet. And as you can see, for all of these foods, our electrochemical measurements track very well with the colony counting, but one thing you'll notice is that our error is order of magnitude lower. So ours gives you much more precise measurements. And so what we've been able to do here is to use the inherent activity of those E. coli to incorporate that boronic acid amino acid to develop an easy and disposable platform to capture and detect pathogenic E. coli. Our limit of detection for this is between 12 and 15 CFU per mil, which, as I mentioned, is several orders of magnitude below the infectious dose. And importantly, we only detect viable cells because we require that active peptidoglycan remodeling to incorporate that amino acid. And from this, we can detect endogenous E. coli in food with less inherent error and several days faster than the current gold standard method. So now we're going to change directions a little bit. And we're going to talk about detecting viruses. I'm sure, yeah?
Oh, wait, so you said it's several days faster than like the current state of the art, so like how fast? Just
So our whole workflow is 2 hours, start to finish.
Cool.
Yeah.
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 With the traces that you increase with the E. coli be detrimental to other cells?
That is a great question. And boronic acid actually degrades naturally pretty quickly. So it's actually pretty harmless.
Okay.
Yeah. Okay, so the next project I'm going to talk about is some of the work from an awesome postdoc in my lab, Arjan, who's in the audience today. And she developed a very creative workflow to detect viral genetic material for both HPV and HIV. So the reason she was interested in detecting HPV is because HPV infections are one of the leading causes of cervical cancer. And importantly, cervical cancer is much more prevalent in low resource settings. It's the 4th most common cancer in people with a cervix, but over 80% of cases occur in low resource settings and developing nations. And current diagnostic methods, which are generally based on visual inspection, are very insensitive, obviously, right? Visual inspection is not great and highly dependent on who's performing it. And so what she was able to do is to develop a workflow that had inherent signal amplification and selectivity by combining basically amplification methods for nucleic acids with CRISPR-Cas12 activation and endonuclease activity. So this is the overall workflow. It's very complicated, but we'll break it down and talk through it. So fundamentally, you have a virus that has genetic material. The first step is to do amplification. We were using LAMP, which is a single temperature method, so it can be done without the need of a PCR machine. We can then specifically activate this Cas12 enzyme, which is a non-specific endonuclease. So once this is activated, it'll chop up anything that's on our electrode. And so that's how we get detection. If this is activated, it'll remove our DNA, and we basically see this on signal turn to an off. If our Cas12 is not activated, then our DNA stays on the surface, and we see the signal maintained on. So let's start with the electrochemical component. How do we modify a surface with DNA to get this specific signal? So the first thing Marjan did was develop these really, really cool, inexpensive electrodes that require only about 16 cents worth of gold and 15 cents for the entire device. And these are based on gold leaf. It's actually the same type that you find in very fancy foods. And you can see here that she basically makes a sticker out of it and can then immobilize it on transparency film, which for those of you who are my age was what they used to use for overhead projectors when those were used for classes. And you can see she makes lead connections with foil, and it's very inexpensive and easy to make. So now, how do we attach DNA to this specifically and get an electrochemical signal? So DNA can be modified with a thiol. This is just through chemical synthesis of this biopolymer. And that thiol readily reacts with gold to form gold thiol bonds. So we just use self-assembly to immobilize that DNA. On the other end of the DNA, we have a redox active molecule. In this case, we use methylene blue. So if that DNA is tethered to the electrode, we'll see a signal at the potential at which that methylene blue probe is active. So we can see that by square wave voltammetry here. As we scan across different potentials, we see a signal from that methylene blue from the DNA immobilized. And as you would expect, as you vary the concentration of oligo from 0.4 micromolars down to 0.004 micromolars, we see that signal decrease. And what that means is we can tune the amount of DNA on our electrode based on how much we have in solution to start. So we have DNA on our electrode. It gives us a signal, right? How do we turn that signal off? So Cas12 is a CRISPR-based enzyme that basically recognizes a specific nucleic acid sequence. So if we have our viral sequence, it'll interact with the guide RNA and activate this. And then Cas12 specifically is a non-specific endonuclease. So once it's activated, it'll chew up any DNA nearby. So we don't need to have a specific sequence on our electrode to get specificity. The specificity comes from the activation step. So what does this look like? If we have our DNA immobilized on an electrode and we have non-target DNA for that Cas12, the Cas12 remains inactive, and we see that square wave voltammeter pre-Cas in gray and post-treatment in red directly overlay. In contrast, if we have our target DNA, it activates that Cas12, which chews up the DNA on our surface. We no longer have a redox probe close to the surface, and we see that signal go from on to off. Does that make sense to everybody? Excellent. So we can incorporate specificity and show specificity for HPV by looking at two different HPV types of HPV, HPV16 and HPV18. So if we have the proper guide RNA for HPV18, we can see that we don't see a response from 16. So basically the signal ratio here is before to after. So if we see a decrease, that means it's present and it's cutting. So HPV18, for the 18 guide RNA, we see turn off. And this is just our no template control. So this is just no DNA added to make sure we're not getting non-specific activation. And if we swap that guide RNA out for HPV16, we see that turn off only for our 16. Does that make sense to everybody? So we get specificity through this Cas enzyme. But often with viral detection, we need to detect very low copy numbers that are nearly impossible to detect without amplification. So next we incorporated a LAMP amplification step. So LAMP, or loop-mediated isothermal amplification, is basically a nucleic acid amplification method that happens at a single temperature. And all you really need to know about it is you design the series of primers that enables this dumbbell structure to form, and then you get exponential amplification. So basically at a single temperature, you can amplify your nucleic acids exponentially. And normally, for those of you who have done PCR, you have to use a fancy piece of equipment such as a thermal cycler. These cost thousands of dollars. Because LAMP happens at a single temperature, you can do it with a resistive heater. But one of the downsides to LAMP, and why it's not used more prevalently, especially for sensing in low resource settings, is that you get false positives. So normally when you think about detection from LAMP, you think about detecting something like a labeled primer. And what that leads to is if you look at this DNA gel here, you can see that For the HPV-18, which is what we were amplifying for, we see amplification. But for this HPV-16, we see this false positive. Importantly, we also see that for our no template control. And so what this would mean is if we didn't have additional specificity, you would often get false positives in your diagnostics. But when we look at that same set of LAMP amplification primers with incorporation of the Cas12 component, so by incorporating that activation step for specificity, we no longer see those false positives. So here we get 100% specificity for that HPV-18 over the HPV-16. So not only has this enabled us to incorporate amplification, but it's also decreased inherent challenges with LAMP. So now we have our whole workflow. We have amplification through LAMP. We have activation of a specific enzyme that then shoots up DNA on our electrode to give us a signal. Can we do this in clinical samples? So Marjan was able to obtain cervical swab samples from several different patients. And as you can see here, her detection has 100% sensitivity and 86% specificity. There was only one false positive here out of all of these samples. And so what she's been able to do is by developing these gold electrodes that are inexpensive and easy to fabricate, she can combine these with other techniques for both amplification and specificity, so LAMP for amplification and Cas12 for specificity, to detect clinically relevant viral loads from actual clinical samples. Any questions on this? Excellent. Then we can go on to the last story that I wanted to talk about. If we're doing good. Yeah.
Yeah, just given the necessity of like proteins and synthetic how do you get your Cas12 for this system? Is it like monoclonal or sorry, I'm not great at proteins, but I assume it's expensive, right?
It is actually not expensive as far as proteins go. You can order it from Sigma Aldrich, so it is viable, or you can express it.
I'm just curious what like how the final cost compares once you include all those other things. Like, you know, obviously the electrode is cheap, but yeah, the fact that you have to put DNA and have these proteins and stuff on it. How does that compare to like a traditional assay?
Yeah, that's a great question. It is actually, the LAMP primers are very inexpensive, and then the cost is still on the order of like dollars per reaction.
Okay, and how much is like a traditional detection?
The commercially available kits can be roughly like $13 a test or more.
Cool. Thanks.
Excellent. Thank you. So the last story I wanted to talk about is how we can detect chemical contaminants in the environment. So specifically, we're going to talk about organophosphate pesticides. These make up more than 50% of chemical pesticides. And importantly, they have the same mechanism of action on your bodies as chemical warfare agents, such as sarin gas. It's estimated that there are about 385 million annual unintentional poisonings every year, leading to 11,000 deaths. And I also want to point out that this is a highly kind of class, I don't know what the word I'm looking for is. It's based on class and based on the country you live in. So the more agricultural workers in your area, the higher likelihood of exposures, and additionally, low resource settings and also developing nations use these pesticides at much higher rates than they do in developed nations. However, I do want to note that they do still use these pesticides in the suburbs in the US for mosquito control. So if you live in a suburb of Boston, you've probably been exposed to these. So what we wanted to do is to develop a single system for both detection and degradation of these organophosphate pesticides. And the way we did this was by developing an engineered microbial mixture where one microbe degrades these compounds and the other detects the degradation products. And this work was performed by an awesome postdoc in my lab, Amruta, who's now at Dartmouth as a research associate, Rachel, who is a UROP in our lab, and Aaron, a grad student. And as you can see, organophosphates, this is their structure. So sarin, soman, and cyclosarin are 3 prevalent chemical warfare agents. You can see the organophosphate groups on here, and this is the structure of the 2 main pesticides that are organophosphates, paraoxin and parathion. You can see that these also contain those phosphate groups attached to the organics, and they mostly just vary in these small side chains here. Importantly for our purposes, both of these degrade to the same product, which is p-nitrophenol. So you'll see me reference this a lot. So as I mentioned, these pesticides have the same mechanism of action as chemical warfare agents. And what we actually care about is that they can be degraded enzymatically. So we can use specific proteins to take those to PNP or paranitrophenol. And so the way we did that first step is by scaffolding those enzymes on the surface of E. coli. So what we can do is we can put a plasmid in E. coli that has a specific tag that tells it to express that protein on the surface. What we also found was that if we lyophilize those E. coli, so they're no longer viable, we still get enzymatic activity. So you can think of this as just a biological scaffold for the enzyme we care about. And so what this looks like is we can detect, so PNP or p-nitrophenol is UV active. That's one way to detect it, but unfortunately the detection limit is high micromolar, as you can see here. But as we increase concentrations of paraoxin, we do get a linear response to the degradation product. So that means that our enzymatic reaction rate is not our limiting step here. Similarly, as we increase the number of cells, we can increase the initial rate of reaction, meaning we can tune this degradation for whatever application we want. So we can degrade it very well, and we can tune the degradation. But how do we detect it? So we developed this genetic circuit. So we have these cells called Shewanella, and I'll talk more about why we chose those specific ones later. But basically what we can do is, how many of you have seen this type of expression before? Nope, okay. So DMPR here is what's called a transcription factor. This is a protein that when it binds to a certain target molecule, in this case, our PNP, it changes conformation and binds to DNA and tells that cell to turn on or off a gene. So this is how we're controlling detection. Here we have under that control GFP, which is green fluorescent protein. So what that means is if we have our transcription factor and it's activated, we get GFP expression or green fluorescent protein expression. What that looks like is we get bright green cells. So if we have that small molecule that turns on our transcription factor, we see green. Without it, we don't see any. And as you can see here, we get very high specificity with this transcription factor. If we have the cells we're looking at with PNP, which is the degradation product, we see significant turn on. This is just fluorescence on the y-axis. If we have the starting material, paraoxin, we see no response, which means it's responding specifically to the degradation product. And if we don't have cells that have this specific plasmid in them, we don't see a response either. So we know that our genetic response is very specific. But obviously fluorescence is challenging to do readout in the field, right? You don't want to have to bring fluorimeter or have to send samples back. So we wanted to basically transform this into an electrochemical signal. How do we do that? So Shewanella are these really cool cells. Basically, instead of respiring onto oxygen, they can respire onto metal oxides. So when we breathe, we put electrons into oxygen, right? That's how we basically respire. Well, these cells can respire oxygen, which is why they're called facultative anaerobes. But if there's no oxygen around, they can actually secrete electrons out of their cells to reduce metal oxides in solution. And in the lab, we can use an electrode as a proxy for a metal oxide. So we can basically get a response from these cells in the lab by poisoning an electrode at the same potential that you would find a metal oxide. We can see this on the right here. We have the time on the x-axis, and this is just current generation. And just from a biofilm of these cells, we can see current growing in to about 20 microamps per centimeter squared. But generally cell membranes are insulating, right? So how do these cells actually secrete electrons? They have this really cool set of proteins between your inner and outer membrane called the MTR pathway. So in the inner membrane, there's this one protein, ChemE, and that transfers electrons through the periplasm to MTRA, which interacts with MTRB, MTRC, and OMCA to actually get electrons from the inside of the cell to the outside. And what this means is that by controlling these proteins, we can control whether or not those cells generate current. So what we found is that the best protein to use for that is ChemE. That was that one on the inner membrane. So if we knock that out and then put a plasmid in that has that complemented dectin with that DMPR, so remember, DMPR is everybody's favorite transcription factor, responds to PNP. Now, instead of turning on something that leads to fluorescence, we're turning on something that generates current. So here's our electrochemical bioreactor. You can see here, it's pretty simply just a jar with a carbon cloth electrode, and then we have a silver chloride reference and a counter. And we can see that if we have those cells with 20 micromolar of p-nitrophenol, PNP, we see current growth. If we have the Shewanella with just the peroxin, the starting material, just as we saw with GFP, we see no current. And if we have this Shewanella only or PNP only, we similarly see no current. So we've now converted that fluorescence genetic system into an electrochemical system. But can we combine them? So I showed you at the beginning that we can degrade peroxin using these E. coli scaffolds. And I'm going to call them scaffolds, and I haven't called this a co-culture because these cells are not viable, which means we can avoid one of the key pitfalls of many co-culture type systems, which is having to keep two different cell types alive. So all we have to worry about is the Shewanella. These are basically just scaffolds for the enzyme. So can we combine those with Shewanella that we've engineered to be PNP responsive to both detect and degrade in a single system? And the answer is yes, we can. So shown on the left here is just the total charge. So that's basically just the area under that current graph that I showed on the last slide. So if we have both cell types plus peroxin, we see a very large response. If we have just Shewanella, we see a very small response, and we think that's because of some kind of background degradation of that organophosphate pesticide. If we just have the cells or we just have the peroxin, we don't see any current. And as you can see, this total charge grows in over time. And importantly, what we see here is there's a dependence on the total charge generated on the input amount of peroxin. So not only can we detect this in an on-off sense as to whether or not it's present, but we can actually quantify how much peroxin is there with significantly higher sensitivity than that UV measurement I showed you at the beginning. We are orders of magnitude lower in concentration than we can detect with UV vis. But is this selective and specific? If you have a biosensor, you need it to be specific, right? Well, we looked at a bunch of different small molecules that you would expect to be in biological matrices. Things like dopamine, hydroquinone, and 4-methylcatechol. And as you can see here, our transcription factor was very specific for PNP. We don't get off-target turn on. Additionally, we can basically show that we can degrade any of those compounds I showed at the beginning and get a turn on signal. So peroxin, methyl, and parathion, which all degrade to PNP, we see that turn on. For maloxin, which is a different pesticide that doesn't degrade to the same product, we don't see turn on. So specificity for the degradation product and specificity for the degradation. So what we've been able to do here is to develop a kind of cell mixture system that allows us to both detect and degrade these dangerous pesticides. And because our E. coli are not viable and basically just serve as a biological scaffold for these enzymes, we only have to keep one cell type alive, which circumvents many of the key challenges with using microbial co-cultures. Importantly, we can detect these organophosphate degradation products at sub-micromolar levels, which significantly outperforms both colorimetric and fluorescence sensors with much simpler instrumentation. So I told you a lot of different stories today about how we can use the inherent activity of everything from small molecules to whole cells to improve our ability to detect dangerous contaminants ranging from E. coli bacteria to HPV viruses to chemical environmental contaminants. And so with that, I'd like to thank my awesome group, my postdoc, Dr. Gong Khan, and Marjan, who I talked a little bit about her work today, my 3 grad students, Alex, Cindy, and Dara, and our amazing group of undergraduates. I'd also like to thank you for your attention. And as the newest member of PPSM, I'm excited to recruit students. So if you're interested, email me. And with that, I will take questions.
Thank you so much. So I know we had already a few questions, but the floor is open. Yep.
Yeah, I have a question about
Can you speak a little bit?
oh yeah. Well, first of all, sorry, great, pretty cool. But I have a question about the use of impedance spectroscopy and how adding like and how increased amounts of E. coli change the charge transfer resistance. I'm kind of curious as to what kind of physically happening. Is it just that the electron transfer towards the ferrocyanide is getting blocked?
Yeah,
And,
that's it. Cells are big. When they bind to your electrode, they block access of the ferrocyanide to your electrode. So it's both electron transfer hindrance and just physical diffusion limitations. That's a good question. Thank you.
So you talked about accessibility to low-income people a lot. So what exactly is like the stability of these sensors? Like if you wanted to like create one and then send them somewhere?
Yeah, that is a great question, and it is highly dependent on the sensor. So for things like the Cas12, unfortunately, we still need cold chain for that because the proteins have to be stored cold. For the first project I talked about with E. coli, those are just chemicals. So we know we can dry down those electrodes and they're stable for weeks. We haven't tried longer than that, but basically that we would envision being a kit where you get a powder that you add to your sample, and then you add that to your electrode, and then you do detection. With the third one, we know that the E. coli are stable indefinitely. We know that we can get viable Shewanella reconstituted from lyophilized cells. So we think that we can dry those down on an electrode and basically deploy that for bioreactors, with the idea being to circumvent the cold chain again, because that's the biggest limitation for a lot of access points.
Cool, thank you.
Class,
Yes.
is there any reason why you want to keep the OPH on the surface of the bacteria?
Yeah, that is a great question. And OPH is both toxic to bacteria, and also we are not sure if lyophilization would maintain enzymatic activity if the enzyme is inside the cell as well. So basically you circumvent the need for the chemicals to actually enter the cell. So you're no longer limited by diffusion into the cell. And we know that they remain very stable, and it decreases the amount of basically effort you have to put into taking care of them. Those cells are indefinitely stable as a bioscaffold. So what we think of them more like is something like a polystyrene bead, where you would put your protein on it, except this, as a grad student, all you have to do is add a small molecule to your cells. You don't have to isolate the protein or purify it or anything like that. So it also lowers the cost.
I have an anecdote that mixes your gold things with the nerve stuff. So there's this one of these fishes, the puffer fish, has that one of those nerve agents, and in Asia, chefs become experts on what you can cut and what you cannot cut, and apparently, like, I'm from Mexico, so in Mexico will be like to the pig, you're not asking for the cheek, but here it's like, oh, if you can just give me a little slice of that stuff, that will numb my mouth. Sometimes they go too much and they die, but actually when they gave it to us, it was so special. Actually, the fish doesn't taste as better. I like more tuna, but I shouldn't say this one. But it comes with gold leaflets, like what you said about those fancy foods. I was like, oh, I'm eating gold. It's like very, very, very special.
Yeah, that's a running joke in our lab that we need to make something for a group dinner that has the gold leaf on it so we can all try it.
Any other question? Oh yeah, there we have. We're going.
As far as impedance spectroscopy, what I know typically when we do it in lab, we have a very large percentage that. How physically large of a system would you be able to run the frequencies that are relevant for your system? I'm just wondering if like there's any like biologically or make it portable.
That is a great question because potentially sets and potentially sets that are capable of impedance are only about 2 years on the market. So within the past 2 years, there are now potentially sets that are about this big that interface with things like a cell phone or a tablet that can do impedance in the range of about 0.1 hertz to about megahertz.
The reason for bacterial specificity with E. coli was a high rearrangement rate of the peptidic glycan layer. How would you, is that unique to E. coli or like do other bacteria present that same feature? And also like when comparing gram-negative E. coli versus gram-positive Staph aureus, like how would you like, differentiate between those two kinds of bacteria?
That is a great question. So in terms of the rate, so what we found is basically all viable cells will incorporate that, all viable microbes that have peptidoglycan walls will incorporate this. It's very rate dependent. So you can develop this as a sensor for other bacteria, but E. coli tend to have the fastest rate, which is why they're usually used for this reaction because otherwise, so Staph aureus you can detect with this method, but it takes about 24 hours and nobody's got time for that. And so I think the main differentiating factor is the rate rather than the structure of the cell, but that's a great question.
If there's no further questions, let's give a big round of applause to